is online, okay? So Goutham will, from Aarhus in Denmark will present Meta-GWAS for mastitis resistance. So Goutham, the floor is yours.
Thank you, Maripari. Yeah, thanks. Could you hear and see the right slides? Could you confirm? Yeah. Okay, good. Thanks for presenting this, the GWAS part in work package 4. And so basically, we were in charge of taking this mastitis, which is under the health conditions. So this is also, again, a contribution for different partners. And then we do analysis together and finish this study. Yes. So of course, the mastitis is not a new issue. Is a long time, both for the economic point of view, antibiotic use, animal health and welfare. There is a genomic selection going on, but there are not that effective because of the low heritability of the trait and unfavorable genetic correlation with the production trait. And many countries, of course, they use the proxy like somatic cell scores, but which is also not perfectly the same trait because the genetic correlation is not 100%, so around 70%. So one of the way to improve the genomic prediction will be to identify if we could get some major effect loci which are affecting mastitis. This is also another advantage will be that this kind of information could be transferable to other related population or breeds. And finally, of course, we are interested to understand the genetic architecture, underlying biology. So if we know the major genes which are affecting, then we can know, okay, what should be targeted, maybe for therapeutic reason, but also how to break this unfavorable genetic correlation with the production trait. So many of the partners were doing this GWAS for the mastitis-related phenotype individually. So in this BOBRAC project, we decided to combine them and do the meta-analysis. The reason being the individual analyses are less powerful because we know the most of the loci have a very small effect to the total phenotypic variance. And also, it's difficult to fine map the loci or the genes because of high LD in the cattle within population. So that will be one advantage when we have multiple breeds, multiple population, we will be able to probably break that LD. And then that was the reason or logic to start the Meta-GWAS to address some of those challenges. So the total population, we have more than 30,000 bull data. These are all progeny tested bulls and also around 120,000 cow data from individuals. So this was brown Swiss and original brown bay. The Holstein was, of course, the major population, then Jersey, Nordic red cattle, Montpellier, and Normand. And as just presented, so what we decided that, okay, we'll do the individual, each partner will do their own GWAS within population because we can't access the basic phenotypes and all the original data. But we, of course, decided what will be the analysis pipeline. So basically imputing everything to 1,000 bull genome. And then using so two phenotypes, clinical mastitis and somatic cell score. These are the two traits, but of course all partners didn't have both. And then using a mixed model-based approach for GWAS. And within populations and for the trait, we found between no genome-wide significant to very large number of genome-wide significant SNPs. And then, of course, because we use the phenotypes, so which has been in a different scale, maybe in different, and also the different kind of proxy phenotypes, regress group or daughter LDPS and DOIDs or those kind of different. So then we also standardize the estimate of the effect based on the genetic variants within population. So that was the data, the summary statistics we got together, combined them. So the pipeline I will explain a little bit here. So we got the summary statistics. So SNP effects, standard error, p-values, allele frequencies, etc. Then we did some quality check because it could be that different allele are scored as an effect allele. So then we can have a different effect. So some kind of quality control is further required for when we do meta-analysis. And of course, initially we decided to use metal, which is just Maripari presented, but I will also present that we use this meta-analysis, which is MR mega. Here it also include, because in the metal, the fixed effect model, we assume that all the variants are segregating in all the populations or similar effect, but these cattle breeds, because of intensive selection, the allele frequency could be quite different, so that the power of estimate or identifying the loci are not same in all population, or in some population maybe this is fixed, so this allele is not segregating. And for multi-trait meta-analysis, we also decided to use another approach, which is called multi-trait analysis of GWAS. And here the difference compared to the other one is that this method approach can take into account the overlap of the samples. And because we have the clinical mastitis and somatic cell score, and both are measured on the same population, or some cases in the cows, the same individual. So there is the overlap of the sample. So that is to especially to account for that. But of course, when we do separate analysis, we get a lot of different kind of SNPs, and that's the complexity of the GWAS, that we not necessarily all approach will lead you the same lead SNPs. But we try to combine from the different approach. So what are the common or intersection of the SNPs, the genes and other things. And then we did a lot of post-GWAS analysis. So gene-based association analysis, MAGMA, effect prediction. We looked into the enrichment of the genomic features. This is specifically if there are some CNVs which overlap with our QTL hits, and try to use this cattle GTEx data to see the overlap with the trans and cis QTLs. And based on all those analysis, of course, we try to come out or propose some candidate genes or candidate specific variants that are possibly causal. So to quickly show the different kind of the QC we said, of course, we check the allele frequencies and with the check with the 1,000 whole genome data, but also across different populations, that looks reasonably well. We also looked if there is an inflation because of the population structure. That was not the case, but of course, some populations there is a depletion, but that could be because we are this mixed model analysis. If we fit the SNP and both in the relationship matrix and as a fixed effect, we could see our estimates could be conservative. We also looked into the effect estimates and the p-values reported and p-values, and of course there was, and most of in all the populations we didn't see any issue with this. So coming to the very summary, I will not go because there are a lot of results. So just a summary of what we found, that we found 15 independent association regions for clinical mastitis, 22 for somatic cell score. And if we look into the QTL database and published literature, so many of them are new hits. And from the gene-based analysis, we had 64 genome-wide significant genes. And we also found that this MR mega approach has a better power for Meta-GWAS in case of cattle compared to the metal, because if you do the metal, we only find this big QTL. Probably many of you know that on chromosome 6, but most of the other one are because segregating only as not all populations. So we didn't say so because that was one of the recommendations from this analysis, or our finding is that MR mega approach is better when we do it GWAS. So I'll also talk a little bit about one of the CNV, which was Lee et al. Earlier reported as a possible causal mutation on chromosome 6. That's the very major QTL reported in many populations in Holstein. So we also tried to do that in the Danish Holstein, because from the sequence data, we tried to see, okay, if this CNB is segregating. And we saw that, of course, it's segregating in Danish Holstein and Nordic red population, but not in Danish Jersey. And if we just see also the QTL signals was significant in Danish Holstein, Nordic red, but not Danish Jersey. But this could be, of course, the Danish Jersey population is small, but also it confirms that possibly this CNB is the causal mutations that is giving this variation in the mastitis resistance in cattle. But just to go into a little bit the deep detail how complex this region is, because this is the CNB, the black, and the blue box are the different blocks of many significant variants. We see the lead SNP for the clinical mastitis is located here, for somatic cells count here, and then of course then there is also a very strong candidate mutations located. So again, though it here, and this is a very high LD region, so it's difficult to break that LD here. So just to show you that the complexity is, even if with this huge data set, by it's not always easy to resolve what is the causal mutations for that association signal. But of course, large number of data, multiple populations, and these annotations that helps to find out or zero down some possible causal mutations. We also looked, of course, in many of the times, what is the problem is that when we report a QTL or associations, this is not replicated in other population. But in this case, we also tried to a relatively big study from US, and some of those chromosomes we have pointed, so the signals overlap. So possibly that the reports what the association we are reporting here, they are real and not the false positive ones. So that confirms. Of course, we did a lot of post-GWAS analysis. So we compare with the cattle GTEx data. So we found that 4 of our significant SNPs actually the cis effect. So majority of the trans effect, but again, that's the complex. We need to look into a little bit more. Are they real, or there could be some false positive? We also looked into a lot of biological support from them. And finally, we are quite confident about 31 putative causal genes and 14 putative causal variants. But again, saying that, of course, the ideal wouldn't be if we could do some kind of a validation study or functional validation study in a biologically validated, but that's not easy or straightforward to do. So coming to the summary and conclusions, that we see that combining the summary statistics from multiple breeds require applications of appropriate methods, because if you are only using metal for Meta-GWAS, so most of the QTL we have missed. So meta-analysis of summary improves power. Of course, we have identified new QTLs, new associations, and then the biological connections, the annotations that helps us to narrow down considerably from the multiple genes in the association region, multiple causal mutations to few ones to follow up. And this information is also passed on to the work package 7, where they Emra and his team also did to use this for mastitis in Danish Jersey, and he will present, of course, his results that how this informations from annotations and QTLs helps in prediction accuracies. Of course, as I said in the beginning, it's a big group. We work together, so from multiple partners. So, and we have already submitted a paper, and hopefully that will be published soon. So then you can get also a lot of the detailed results from the publication. And thanks all the partners and the contributors for the data analysis and of course finance for of course part of this BOBREC project, but also the individual countries. And thank you for your attention.
Thank you, Goutham. So questions for Goutham? Yeah. your axis.
Juliaxis from IRTA. Thank you for the presentation. It was not clear for me when you talk about the CMV, you test the segregation in the population or the association with the trait?
The CNV we called only from the sequence data. So we cannot directly test if the CNV is associated with the phenotypes. But we test the CNV, LD of the CNV with the lead SNPs, and that was almost completely in LD with the phenotypes.
Okay.
But we couldn't do it because the calling of the CNV is only possible for the sequence data, and that's only very limited number. So we couldn't do a direct test.
Yes, and that was my point, because the breakpoint is really difficult to establish because there is a lot of variability between individuals. So my suggestion is, why not, rather than try a particular SNP, try to haplotype the region, and then with the haplotype, theoretically, you can capture better the signal in the region, including the CMV. So probably it's better to try haplotype rather than a single SNP for association with CMV. It's just a suggestion.
I'm not sure if the haplotype will help here, but what I think, because we are, as you say, also this custom chip. So if we can have some kind of a SNPs or the CNV which can be tagged or genotyped directly, that could be an option, I think, in future. But the haplotype, I am not sure if that will help in this case.
Thank you.
But I don't know. No.
Okay, other questions in the room or online, maybe? No? Okay, so thank you again, Goutham.